now.
Sure, sure.
Hello.
Morning, everybody. I would request everyone to
Someone's
mute yourself.
mic is not muted. Please, I request everyone to mute your mic except Jay. I request everyone to mute your mic except Jay.
Thank you, sir.
Gunia, then Shahu, please mute your mic. I request all others to mute your mic. Okay, thank you.
Thank you, everybody.
Yes. Dr. Jay, you can proceed.
Thank you, Dr. Deshmukh. Respected Deshmukh, sir, Minal Trivedi, ma'am, today is our speaker, Dr. Vivek Parab, and everybody else. Very good morning to all of you. I would now like to request our Gujarat State MSi President, Dr. Minal Trivedi, to speak few words, and afterwards I would introduce the speaker.
Good morning, everyone. Honorable Deshmukh sir, President of MBSi, Jay, Jatiya, coordinator of this event, all the eminent resource persons and participants for this webinar, and today's expert for the presentation. I feel very happy to have an overwhelming response in this webinar series concerning to the speakers ready to participate. And we are very sure that this series will continue throughout the academic year 22-23. So I'm thankful to Deshmukh sir for this, and I am congratulating Jay for this very nice idea to organize this webinar series. Every time we are coming with a new topic from one resource person, and this definitely provide us guidance to the junior students also regarding the opportunity research in microbiology. I again welcome you all, and without wasting time, I request Jay to introduce our speaker and start the session. Over to you, Jay.
Thank you very much, ma'am. Today's our speaker is Dr. Vivek.
Hello, Jay.
Yes, sir.
I want to talk only for 1 minute.
Please, sir.
The reason is that I want to appreciate Gujarat unit of Microbiology Society, including Minal ma'am, including Jay. The reason is that really it is an innovative idea. Young generation from India is doing research work. It is a duty of elder teachers to give wide publicity to them, to give exposure to them, and to appreciate them. We are appreciating all young generation delivering lecture via virtual conference by giving a memento and certificate. So they will remember, they will get inspired, and their research work will be followed by other, because there are many other young scientists in India want to do work with the same line. So really it is an innovative approach, and I again congratulate. I also express my happiness because many senior scientists from India are here to discuss and to inspire, to encourage young generation. Thank you very much. Thank you.
Thank you, Deshmukh,
sir. Sir, one more thing. Apart from appreciating all the speakers, I do appreciate Jay for this innovative idea and consistent coordination of this event. So we congratulate. Even it is required to be felicitated. Jay, you also need a letter of appreciation from MBSi.
Thank you, ma'am.
Thank you.
Actually, I would like to give all the credit to Deshmukh, sir. With his guidance, I think we are moving forward very nicely. Thank you. So actually, I would like to introduce our speaker today. Our today's speaker is Dr. Vivek Parab. Dr. Vivek has done his bachelor's and master's in microbiology from University of Mumbai. Dr. Vivek has completed his PhD from the same university University of Mumbai in 2022. And there is one very interesting thing I would like to mention about Dr. Vivek. Dr. Vivek has qualified GATE exam in 2014 with All India rank 867. Before that, Dr. Vivek worked as an assistant professor at Karmavir Burao Patil College, Vashi, and sir has also worked as Department of Biotechnology, Vidya Vikas Education Society, College of Arts, Commerce and Science, Vikhroli. Other than that, Dr. Vivek has served as a technical assistant, SIES, Institute of Chromatography and Spectroscopy, Nerul. Currently, Dr. Vivek is employed as a technical officer, research and development, at SIES School of Packaging. Dr. Vivek has published more than 6 international research papers with high impact factor journals. Other than that, Doctor has participated in poster presentations and national and international conferences, counting more than 10. Other than that, he has also received more than 12 national and international awards. So in the interest of time, I would like to welcome Dr. Vivek. Please start your presentation.
Thank you, sir. Am I audible?
Yes, loud and clear.
Okay, sir. Okay. It's my honor to present my work in front of such eminent people. I would like to thank Ms. Dr. Jay, Dr. Arvind Deshmukh, sir, and Minal Trivedi, ma'am. So I will start my presentation. Just let me know, sir, if it's visible.
Yes, you are visible.
Okay, sir. Yeah. Okay, so good morning, everyone. The topic of my presentation, the topic of my PhD work is isolation of naphthalene and phenanthrene degrading microorganisms and their applications in bioremediation of contaminated sites. The project was conducted under the guidance of Dr. Manju Furke ma'am in Department of Microbiology, SIES College, Arts and Commerce under the University of Mumbai. Okay, so when you talk about hydrocarbons, you have aliphatic and aromatic hydrocarbons. I focus mainly on aromatic hydrocarbons because, first of all, they are considered as environmental pollutants, okay, by US EPA and PCB. So if we see the sources of aromatic hydrocarbon pollution, there are both natural sources and sources by anthropogenic sources, that is sources by humans. You have industries, domestic sewage, which has been released inside the water, in the water. You have volcanoes which release so much of smoke and all. Then you have wildfires. Then you have small sources. You have smoke coming from vehicular emissions. Then you have tobacco smoke. All these things lead to huge amount of pollution by pHs, and pHs are basically not soluble in waters, so they remain in the sources and can enter the food chain if they are consumed via some aquatic animals, like you have fishes and crabs, etc. And they can enter human body through various sources, like you have skin contact, which can lead to skin cancer. You have inhalation of the gases to lung cancer. You have ingestion through eating or certain foods which have been contaminated by pHs, which can lead to stomach cancer. Now, when I talk about these compounds leading to such diseases, it doesn't mean that a little bit amount can do harm, because if we see, there are many sources like food sources which may also contain pHs, but the concentrations are very low. But if it has been increased than the permissible limits, then it could lead to harm. Now, with respect to some major studies which have been conducted on different samples, like you have soil samples, sediment samples, water samples, fish samples, then you can see these are certain different concentrations of pHs which have been found in such different sources. And these are certain permissible limits which are of pHs which should be there inside water and soil and sources. So if these exceed outside the sources, then it could lead to harm. And if it continues, then it could lead to cancer and such harmful, toxic, fatal diseases. So my focus was on mainly naphthalene and phenanthrene. The reason is because I didn't want to go towards the higher compounds. I initially wanted to do a basic study on basic hydrocarbons. So why I chose naphthalene and phenanthrene only? First of all, naphthalene is basic hydrocarbon having two rings, which is considered as hydrocarbons, aromatic hydrocarbons. Then I focused on phenanthrene, which is a bit more complicated, complex than naphthalene, having three rings. Now you have two compounds basically, anthracene and phenanthrene, having three ring compounds. But the speciality or the difference between these two compounds is phenanthrene has a different angle. That is, they have two regions mainly, that is called as Bay region and K region. Now these two regions represent higher molecular weight compounds. So these both are low molecular weight compounds, and phenanthrene and naphthalene, they represent higher molecular weight compounds. So if we study these low molecular weight compound, you can get an idea of how the organisms would be able to degrade and work on the higher molecular weight compound. Hence, the phenanthrene was chosen, and Bay region and K region are the main region which can lead to mutations or causing compounds inside human cells if they enter the cells. Now, the objectives of the studies were very basic and simple. So, because I wanted to do some ground research and not very advanced one. So, before going to field studies, I wanted to do mainly lab scale experiments. This was the main objective of my main aim of my PhD project. So, first things were hello. So first objective was isolating these compounds from different sources. The sources were like soil sources, water sources, soil collected from petrol pump areas, soil collected from railway tracks. All this soil which was collected were all contaminated by either petroleum products or oil or any other products which can have hydrocarbons in them. The organisms which may be living inside them can be able to utilize these hydrocarbons. Now, the growth was done. The organisms were grown in NS mineral salt medium containing only naphthalene and phenanthrene as the sole carbon sources. They were first done inside a petri plate, and then it were carried on into broth medium. Okay, so next focus on revealing the biochemical and genetic controls. That is, what kind of byproducts or metabolites are produced during the degradation of pHs, and also which genes may be present inside these pH degrading organisms, isolates. Later on, to calculate the degradation of pH using chromatographic analysis to find out how much amount of pH has been degraded and remaining inside the media. The main objective was to develop a small biofilm bioreactor, a lab scale bioreactor, using these bacterial isolates and forming a consortium of these isolates, and to check whether these organisms can able to form biofilm. So these biofilm forming consortia can help in better degradation of pHs at different concentrations of pH, and utilizing them in degrading wastewater inside a confined bioreactor, a controlled environment inside the lab, and hence it can be further scaled up future field studies later on. So I focus on two degradation pathways. So there are two main different degradation pathways of pHs. That is ortho cleavage pathway and meta cleavage pathway. In autocleavage pathway, you have degradation of pH to catechol and salicylic acid, and further different metabolites that could lead to this metabolites being used in TCA pathway for bacterial growth. And the byproducts from these two pathways, like metacleavage pathway and autocleavage pathway, will be carbon dioxide and water, which are comparatively very clean and no toxic once the degradation has been continued. Okay, so these two pathways are focused upon to check different metabolites which have been formed. On the previous studies, I found out that it can be possible to determine the production of catechol inside the when the pHs are degraded by the bacteria. So my focus went on to determining catechol inside this degradation pathway by organisms. So I focused on metacleavage degradation pathway. in metacleavage degradation pathway, the catechol is converted using the enzyme by the enzyme catechol 2,3-dioxygenase by the organisms, and it is converted into 2-hydroxymuconates ML. Now the trick is to find out when the organisms are producing this 2-hydroxymuconates aldehyde. So the reason is because when organisms produce this compound, the final product, the organisms turn reddish in color. The pigment, no, it's not a pigment; it's actually a chemical reaction. So the colonies will form red color colonies, or the media will get red color to brown to amber color media. So my aim was to determine, my objective was to determine whether organisms are producing catechol 2,3-dioxygenase, and also to find out whether the organisms are able to produce catechol. Okay, since they will produce catechol, they will produce this enzyme, and hence they will produce hydroxymuconic in the presence of catechol dioxygenase. And later on, these organisms can move towards the final products, which can lead to metabolites which can be used by organisms in TCA pathway, in the degradation, in the basic growth metabolite pathways. Okay, so I used two methods. One of the method was Franklin's method, where if you add catechol, okay, if you add what do you say, catechol, standard catechol on the colonies. So what this method determines is if the catechol, if the producing catechol 2,3-dioxygenase enzyme. So what you do is you add cyanocatechol on the colonies. So if they're producing this enzyme, this catechol which you're adding on the organisms, they will they will use this enzyme to convert this catechol into 2-hydroxymuconic semialdehyde. See, the organisms, when they produce inside the media, it's different. And when the organisms, when they are given by the catechol by yourself, is different, because you provide them higher concentrated catechol, which gives them bright red color. And hence, what I found out that organisms, wherever I added catechol, the organisms colonies turned red in color, which indicates that production of catechol dioxygenase was there. And hence, there was another method to find out the production of catechol, that is Arnow's method, where you can add nitrite molybdate reagent in the media, and when the organisms are growing, when the growth is happening, this nitrite molybdate will turn not only the colonies but also the media into golden yellow or amber colored when catechol is being produced. Okay, so I found out catechol production and the production of the enzyme is catechol dioxygenase. Now these are all these mostly were the qualitative estimation, and I also performed the quantitative estimation to find out how much amount of catechol was produced. And I found out that when the organisms were degrading my compounds, that is PHs, naphthalene and phenanthrene, they were able to degrade the concentrations of phenanthrene and naphthalene in a mixture. Okay, they were not provided as individual components, but as a mixture, naphthalene and phenanthrene both. The degradation was up to around 1 week or 1.5 weeks. Okay, so what I found out that when a catechol was produced, it was degraded at concentration at around the catechol was produced at around 48 hours, and later on the further degradation took place. Okay, so these were the concentrations of catechol, and when you know that going in the presence of pH, that means they're utilizing pH because there is no other carbon source. And also you know that when the organism is producing more catechol, it is degrading more amount of pH. So it's kind of relatable that both organisms they're degrading pH also and they're producing catechol also, so relatable. Okay, so further on I moved towards increasing the concentration of naphthalene and phenanthrene inside the media. So since they were given as a mixture, the concentration was raised from 100 ppm towards 500 ppm, and later on when I developed the consortia from the organisms, I added more amount of concentration of pH, that is 1000 ppm per pH, that is naphthalene and phenanthrene. So the organisms which were able to degrade pHs at a concentration of 500 ppm were only used for consortia development, because lower the organisms which, and also they were the only organisms which were identified, because I found out that only these organisms can be used for further studies which were able to degrade at least till 500 ppm. Later on, these bacteria, these isolates were used for development of consortium, and this consortium was used for degradation of pH at a concentration of 1000 ppm. Later on, I checked whether these organisms were not only able to degrade at 1000 ppm in the form of consortia, but also more than 1000 ppm if they were able to degrade. So from 500 ppm, the concentration of pH, that is both naphthalene and phenanthrene inside the media, was increased to at least 2000 to 2200 ppm. The reason I reached only till 2200 ppm is because that's where the growth stopped. So the MIC was found out. So the results I will show you in the later slides. And these organisms which were able to degrade at higher concentration, like 2000 ppm or 1800 ppm, were only used for bioremediation studies in bioreactor. So these were the results which I obtained from the different isolates. So initially, I got around 60 isolates. From those isolates, I chose 10 isolates which were able to phenanthrene and naphthalene individually as a mixture in a media at a concentration of 500 ppm. So these are the results which I obtained from chromatographic analysis. So I cannot show all the chromatograms, so I displayed in that form of table. So these were the results. The organisms, the isolates which were VP50, VP29, and VP30 were able to degrade naphthalene at a rate of around 900% and 99%. Okay, so initially rate was very low. It was around 60%, 70%, somewhere or 80%, 30%. But some isolates were able to degrade at higher rate. Okay, and in the presence of naphthalene, phenanthrene was also added. So not only the organisms were degrading naphthalene, they were also degrading phenanthrene. So the percentage of phenanthrene was comparatively low than naphthalene degradation. So as you can see, when the naphthalene degradation is very high, the rate of degradation of phenanthrene was comparatively low. As you can see in between 20, the degradation rate of naphthalene was around 89%, but the degradation of phenanthrene was only 2%. So this way, there were organisms which were not able to degrade both efficiently. Some were able to degrade both efficiently, but lower amount of phenanthrene was degraded. Now, if we compare these organisms, these are isolates with some other isolates which were initially studied in earlier studies, what you can see is the well degrading organisms also were able to degrade the phenanthrene at a concentration of around 200, 250, maximum at 500 ppm, which I also was, my isolates were also able to degrade. The days which my organisms took were 6 days. Okay, but the other studies showed that they were degrading not in 6 days, not only in 6 days, but in 23 days also. Some were degrading at 15. So the percentage degradation was somewhat same to my organisms, like some were degrading at 87%. If they were degrading phenanthrene at 85% in 6 days, the concentration was low. If the concentration was high, the days taken by the organisms for degradation of phenanthrene was low. So my aim was to get these organisms in a consortium form and degrade even higher concentration of pHs at least lesser days than 15 days or 14 days or 2 weeks at more percentage. Okay, so more concentration of pH, lesser days, and higher degradation rate. Later on, the isolates which were able to degrade 500 ppm pH phenanthrene naphthalene were identified, and sorry for the strains I have not mentioned as italic form, but so I should have named as an italic format. Okay, so the organisms which were identified were the species were Chrysobacterium, Chrysobacterium. Two Chrysobacterium species were there, one Strenotrophomonas and one Pseudomonas. So everywhere where you find out any degradation mechanism, Pseudomonas species always there. These isolates were the sequences of these isolates were submitted to NCBI, and these are the accession numbers of Now, these organisms were utilized for development of consortia for degradation of further concentrations of pHs. Now, these were the consortia which were developed. Now, normally, what we see is a formation of consortium when organisms are used for development of consortia. You can see only 2 organisms or 3 organisms are showing better results, and if you add more organisms, it's very difficult to obtain them as a giving any synergistic action or additive effect. But what I found out that when I developed my consortia, I used all the organisms which were able to degrade at 500 ppm, and the viable count was checked at every time, and OD was kept constant at 0.5. So what I found out that first thing I want to make clear that when I identified the organisms when I developed the consortia, so the organisms were mixed first and then identified to find out whether actually the consortia, that the cultures which I'm mixing, are present over there and not consortia is not formed by a fluke. Okay, so I checked with different consortia from all the organisms to less organisms. So what I found out that in consortium 5, when these 6 organisms were mixed, there was more degradation rate of both phenanthrene and naphthalene at a rate of 800% and 98% at concentration of 100 ppm each. Whereas in some other cases, I found out some organisms were not able to degrade better in an efficient way, as addition of certain cultures was affecting the degradation of pHs, and they were reducing the rate. Like you can see over here in the consortium 6, addition of organisms, removing of some organisms was reducing the degradation of phenanthrene, which means that you require some other organisms for better degradation of phenanthrene. As you can see, in some cases, degradation of naphthalene was decreased when you add or remove some other organisms. So for better efficiency of organ degradation, you these 6 cultures which were working synergistically and having a good effect on degradation of this degradation of phenanthrene and naphthalene. So if I compare my results with the previous studies of consortia formation and degradation of aromatic hydrocarbons like phenanthrene and naphthalene, what I found out that even in some previous studies, more than 2 or 3 organisms were used. Like in this case, 1, 2, 3, 4, 5 organisms were used as consortia. Other where you have 3 organisms as consortia. Some have Pseudomonas. Most of the studies have Pseudomonas. Some of the studies also have Stingobacterium. Some of the studies also have Sinotrophomonas. So these were the main references which I referred. So you can see the degradation percentage was around 70%. Or you have 98%, you have 85% in 14 days. So, by the way, I want to mention that the organisms the consortia which I've developed was able to degrade the phenanthrene and naphthalene in a span of 6 days. So basically, I focus on the study that the organism should be able to degrade at least in 6 days or maximum 8 days, not more than that. This will be much efficient for me to use this organism for further studies. So you can see the studies which were carried out earlier had degradation happening in 14 days or 10 days. The lowest was 4 days, but the concentration was low, that is 100 ppm. There was also a study where the organisms degraded in 45 days. Though the efficiency was good, 81%, but it took 45 days of degradation and the concentration was just 3.5%. So you can see the consortium which I am using not only degrades individual hydrocarbons, but the hydrocarbons in mixture. So naphthalene and phenanthrene were mixed in a single broth. So it's not just one hydrocarbon they are degrading both the hydrocarbons within a span of 6 to 8 days, and the concentration was as high as 1000 ppm. You can see here it reached around 1000 ppm. It is 750 ppm, but not close to mine. So my organisms are able to degrade a very high concentration. So these were used for further studies. Then I wanted to find out whether what genes were present in the organisms that were responsible for producing these degradation proteins and these enzymes for the degradation. So I focus on three main genes, that is categenes, endogenes, mutagens. Now these genes are found in organisms which can degrade phenanthrene and naphthalene using metacleavage pathway. Now this catechene is, as you can see the name, it is mainly for catechol production, catechol degradation, catechol production during the phenanthrene and naphthalene degradation. Then endogene and mutagens are mainly used in phenanthrene and naphthalene degradation, both phenanthrene and naphthalene degradation. And these are the enzymatic systems which are being produced using these genes, and these are the primers which can be used for finding out these genes. The organisms which are used for my studies were the isolates which I had identified. The representative isolates which I used were Agromyces, Strenotrophomonas, and Pseudomonas, because the genes which I chose were also studied in the species that were Agromyces, Strenotrophomonas, and Pseudomonas. So I wanted to use those genes which can be related to my strains, and also these genes should have been previously either known to be present in these isolates or somewhat studied in these isolates. So what I found out that there were very limited studies which were done on these genes in these species. So I chose these genes for to determine whether they're present in my organisms or not. So what I found out, yes, the categenes, the net genes, and endogenes were present in my isolates. That is, categene in my Agromyces species. Mutagens was present in my Strenotrophomonas species, and Pseudomonas species. The isolate which I have got contain endogene also. So I can confirm that yes, these are responsible for degradation of pHs. Then I found out the MIC, that is minimum inhibitory concentration of pH on my organisms. That is a consortium which I had developed. So I have found out that the consortia was not able. So these are all the studies on consortia and not a single organism. So what you can see is when I increase the concentration till 2200, the growth rate suddenly decreased, whereas when I increased the concentration till when the concentration was low as 500, you can see a good amount of growth, but at higher concentration the growth was decreased. So I cannot use these concentrations for the study, so I chose 2000 ppm mg per liter for further studies in the degradation of hydrocarbons at a higher concentration in my bioreactor studies. So what I thought of doing is optimizing the media. So I chose design expert software for optimization of my media. So these were the factors, these were the parameters which I chose to optimize in the media. So basically I chose different salts concentrations. I also chose different pH and temperature as the factors for degradation, as the factors for affecting the degradation rate. So this was the Plackett Burman design which I was showing you. So those were the different runs which I carried out. Now half normal plot and Pareto chart showed that in the degradation of naphthalene, factors which were affecting the growth rate, that is degradation rate, were pH and H3PO3. Okay, so boric acid. Then for degradation, for phenanthrene degradation, the factors mainly affecting were pH and NH4Cl, boric acid, and COCl2. And also the main factor was Na2HPO4. Over here also Na2HPO4 was showing a significant effect on the growth, on the degradation rate. Then one factor analysis can show you how much, what concentrate, what the value, what is the concentration of Na2HPO4 which has been used in degradation rate. So these are the different values, that is concentration of Na2HPO4 and pH affecting the degradation of naphthalene and also H3PO3. This is the cubic design. Then you have factors like Na2HPO4 and NH4Cl and pH affecting the degradation of phenanthrene. Okay. So these, and this is a cubic design. You can see different, what are the concentrations which are affecting the degradation of phenanthrene. And these concentration was further used in optimizing the media and using these optimized media for further studies. Now the focus was on finding out whether the organisms were able to produce biofilms or not, and also whether organisms were producing biosurfactants or not. Biofilms, as in, for the reason was to form a biofilm bioreactor, so the organism should be able to form biofilm. The biofilm can enhance the uptake, that is utilization of this, not uptake, the utilization of these hydrocarbons in a very strenuous environment. And also biofilms will enhance the uptake of the of the pH, that's naphthalene and phenanthrene. So organisms should be able to produce biosurfactant also and biofilm. So to check the biofilm formation, I chose to standard method, that is LSN Sutherland and also crystal wallet assay using LSR micro titer plate reader. So the organisms, the consortia which I formed, was able to produce the biofilm within 72 hours, and crystal wallet assay was a quantity analysis which helped me to find out how much amount of biofilm is in form when compared with positive control, that is Pseudomonas aeruginosa, which is known for forming. Okay, so the media which I used mainly was MSM media. I also checked the biofilm formation in the presence of urea butane media, in the presence of glucose, in the absence of glucose, at lower concentration of pH, at higher concentration of pH, and these were the different results which I got for the formation of biofilm. And it was compared with the positive control Pseudomonas. I also so I checked the production of biosurfactant using drop collapse assay. So you can see when I added negative control distilled water on the drop, oil drop, there was no collapse of the drop, whereas twin 80 surfactant, when I chose to find out the production, the drop collapse, this was the result. Whereas my organisms, the consortia was able to collapse completely. The oil was completely collapsed, which means there was huge amount of biosurfactant production, and this biosurfactant was helping those organisms to uptake the pH at a very high amount of at a very high rate. That's the reason they were able to utilize these pHs at even the concentration of 1000 ppm or even 2000 ppm. So later on, I designed and developed a small biofilm bioreactor at lab scale. So this is not like those general bioreactors which you see in laboratories, but it was designed by me. So this is a prototype model. So you can add some changes and modify this bioreactor further. So this is the basic structure. You have a 2-liter bioreactor with inlet for water and outlet for effluent. You have air which is been pumped inside continuously. So it can be used as a continuous bioreactor and also a batch bioreactor. You have perforations for pneumatic mixing of pHs throughout the media, throughout the growth media for better degradation. You have a glass door with fixed glass slides. The length of the glass slides can be increased. This was for biofilm formation inside the media. So I know the surface area for biofilm formation is low over here as compared to the bed and beads which have been used in normal bioreactors. But this can be modified later, and this was just a prototype to study how the biofilm formation is there and how the biofilm formed can affect the degradation. Use high concentrated pHs more than 1000 ppm. So this is the actual model which was been developed by our glass blower. And this was used for further studies of degradation of pHs at high concentrated pHs, that is phenanthrene and naphthalene. So these are the chromatographic results of naphthalene and phenanthrene at a concentration of 2000 ppm, where the media was the organisms were growing in wastewater and not the MSM media. So organisms were added in wastewater containing pHs which I added. So it was a kind of simulated environment, but the media was wastewater along with naphthalene and phenanthrene. Little bit of at least 5 ml of MSM was added for initiation of growth of the organisms. The carbon source was only naphthalene and phenanthrene. Organisms were anyways able to produce biosurfactant, so they could uptake the pHs at efficient rate. So you can see this is the initial day study of pHs, naphthalene and phenanthrene. Later on, you can see the naphthalene and phenanthrene were slowly able to degrade at a concentration of 99% to 91%. This was at day 3, and by day 6, the degradation rate was around 99.9% for naphthalene, that it was almost completely degraded, and 92% for phenanthrene. Concentration of 2000 ppm, and hence it was expected that organisms cannot degrade more than what it was degrading earlier. So this was unoptimized media which was added in the wastewater. As I told you, the media was optimized, and little bit amount of 5 ml of media was added inside the wastewater. Later on, the volume of the media was increased from 5 ml to 10 ml. 100 ml of unoptimized media. So the final volume was 100 ml plus 100 ml of mineral salt, unoptimized mineral salt media. And these were the results in the presence of these two components. Later on, I added optimized mineral salt media that was optimized after the Plackett Burman study and all. So here now the contaminated water, the wastewater which I added was around 1400 ml and 100 ml of optimized media, with all the factors which were added later. And the concentration of naphthalene and phenanthrene which was added was same, 2000 ppm, but the rate which I observed was completely different. So earlier, if you compare with the earlier studies, the degradation rate was much higher, and more amount of phenanthrene was degraded at day 3 itself, and by day 6 the degradation rate of phenanthrene and naphthalene was even more increased. You can see the naphthalene degradation was earlier in this earlier study was around 99.9. Here you can see it's 99.87%. For phenanthrene, the degradation was now 97%, 92% for earlier studies. Also, the COD of the water was checked, and it was found to be decreasing, which means the organic matter of water was decreased when the pH degradation was happening. If we compare both optimized and unoptimized media when they were added to the wastewater treatment, so you can see addition of optimized media increased the rate of degradation very substantially, and so these media and these organisms can be used for much further studies. So these were all scale-up studies which were done. So earlier studies which I showed you were done at lower lower volume of the media and growth media, that in the lower bioreactor, that is 1 liter bioreactor, and then it was started from flask to 1 liter bioreactor, and then it moved towards the 2 liter bioreactor which I showed you. So these studies, the growth media, the wastewater which I'm added, the bioreactor which I have constructed can be scaled further from 2 liters to 20 liters and even more. And it can be also used in develop biopit for the studies of pH degradation. So summarizing and concluding my work, I would say that organisms were able to degrade from 500 ppm 100 ppm to 1000 to even 2000 ppm of phenanthrene and naphthalene as a mixture in a single media. They were able to degrade the pHs using meta-cleavage pathway, because there was a production of catechol and catechol 2,3-dioxide time. The isolates which I obtained were Crassiobacterium, Sphingobacterium, Cyanotrophmonas, Agromyces, Pseudomonas. So what I want to mention over here is limited to no amount of study is done on Agromyces species when you consider pH degradation. So I would consider that my work would be a pioneer work for using Agromyces species in pH. You can find out some studies containing Sphingobacterium, Cyanotrophmonas, and Pseudomonas which were used in degradation of pH, but Agromyces is not used much. And luckily, I got these organisms. Even Crassiobacterium can be used in previous studies. So these organisms, when they are mixed in consortium and developed as a consortium, they are able to degrade 2000 ppm. So in further studies, I could study more amount of pHs apart from naphthalene and phenanthrene, more higher molecular weight compounds, and also in a scaled up bioreactor to find out whether they are showing any better degradation at these different scaled up rates of pHs, and also I can find out some more genes and some more metabolic pathway for degradation of pHs. So here I conclude my work. I would like to thank my guide, Dr. Manju Farke, for her event support, and my lab partner, Ms. Lynn Dilemma, who has been always cornerstone during my PhD studies. I would like to thank all the staff members, principal, and from different departments of SIES College of Arts, Science and Commerce. I would like to thank all the departments of Vikas College of Arts, Science and Commerce, because there I initiated my studies when I initiated my PhD studies. When I started my PhD, when I was working over there, I would like to thank SIS Institute of Chromatography, where who helped me understand chromatography and also trained me in GC and HPLC for performing this chromatography studies by myself. I'd like to thank SIS School of Packaging for help for supporting me during my final days of PhD thesis submission. Department of Microbiology, VS Arts, Science and Commerce for not only teaching me and making me capable enough to work in microbiology field, but also helping me during my biofilm formation, biofilm detection studies from using crystal word assay. I would like to thank Institute of Chemical Technology for helping me in determining the genes in the bacteria. So here I conclude my work. These are the references which I use. There's some of the references which I use during my PhD studies. Thank you.
What an excellent presentation, Vivek sir. Very nice.
Thank you, sir.
Thank you, sir. Everybody now, please feel free to ask your questions, or either you can use a chat box.
So Ranjit sir, any questions? So I suppose sir is muted.
Vivek sir, you can stop saying.
Can I say one or two questions, please?
Yes, sir, please.
It was an excellent talk by you.
Thank you, sir.
And I congratulate you for the good work.
Thank you, sir.
Now tell me, what was the role of plasmids in the degradation of phenanthrene and naphthalene?
Yes, sir. About plasmid, actually, I tried to find out the presence of plasmid, but what I found out that the genes which were there in degradation were chromosomal based, because I could not cure any plasmid or detect any plasmid in the organisms as of in my studies.
Sometimes plasmids are difficult to catch.
Yeah, exactly. It's very difficult to find out.
You have to try again. Now you're free from the thesis.
Right.
Exactly. From the time limit.
Yes, I have now time.
If you can report a plasmid, which is there, phenanthrene and naphthalene, your work will be highlighted more.
Exactly, sir. Exactly. I was trying to find out, because those are some plasmids are more they are more responsible for better degradation, because there was so huge amount of degradation, which means there might be some presence of plasmid, but I was not able to find, unfortunately.
The degradation of hydrocarbons is a special function, right?
Exactly, sir. Exactly.
It does not have oxygen. It has got only carbon and hydrogen.
Exactly, right, sir.
There is no oxygen molecule in the molecule.
Right, sir.
So it is well known that specialized function is done by specialized plasmid.
Exactly, right.
And you please change the concentration of the gel. The
concentration
gel concentration is a trick,
Okay, gel. Okay.
Change the concentration of the gel 5%, 5%, 5%, and I'm sure you can catch the plasmid.
Okay, sure. So I'll definitely.
you can publish this work in a very high impact journal.
Okay. Sure. Thank you for the suggestion. I
And
will keep
number
in mind.
two, number
Yes, sir.
two, the biosurfactant you got from which bacteria?
Actually, when I did individual studies for biosurfactant production, it was mainly Chrysobacterium, Pseudomonas, and
Yes.
Fingobacterium. So these were the three organisms which were producing biosurfactant when I did individual studies. But later on, I used consortia for finding out biosurfactant production. So
Okay.
main three organisms were Chrysobacterium, Sphingobacterium, and Pseudomonas. Agromyces organisms, Stenotropomonas was not able to give that much amount of biosurfactant production, but there was little amount of. So I didn't quantify it, but I qualitatively analyzed it.
The Pseudomonas, they normally form rhamnolipid.
Rhamnolipids, exactly, sir.
And the bacillus, they formed lipopeptides like
Right.
surfactant, right?
Exactly.
So did you go further to purify your biosurfactant?
No. I didn't move ahead because I focused on some other parts of the study. But then, yes, I can purify the biosurfactant also.
That is one more exciting work you can do.
Yes, sir, the rhamnolipid. Because I remember, sir, I want to share something because in my MSc studies, my post-graduation studies, I was working on biosurfactant production, and the organism was Pseudomonas only. And there I found out rhamnolipid as biosurfactant. I didn't purify that too, but then yes, it was rhamnolipid from the Pseudomonas which I have got there.
You know, the surfactants have got a lot of applications.
Right.
And you can always use them for industrial application.
Right.
That's why I'm saying you can try later on.
Exactly. Sure.
It was a great talk. I'm very happy. Thank you.
Thank you so much.
Hello, Vivek. I have a question.
Yes.
Which solvent was used for the extraction of naphthalene and phenanthrene after the degradation?
Okay, so I use N-hexane solvent.
Have you determined the recovery rate of that solvent?
It's around 80 to 90% of recovery rate for
Okay.
N-hexane, because other solvents were giving even lesser. So N-hexane was preferred. In case of CCl4, you get more recovery rate, but it's a bit not. It's a bit toxic than your N-hexane. So I use N-hexane.
So okay.
Yeah.
Sir, can I ask a question?
Yes, ma'am. Yes.
Sir, I want to ask on exactly what test means. You said ki MIC you use for the deciding the strain, which is giving better results.
Yeah.
Is that the only criteria to decide the strain?
Besides strain or
for better growth, for better degradation?
Criteria as in, see, the main work was on how much growth rate was observed and how much degradation was observed in how much days. So based on that, I chose the strains. So the criteria was they should be able to degrade high concentration. They should be able to have good growth rate in less amount of time, and also the concentration of pH should be high enough. That's where the main criteria of. Any suggestions from your side? Any other criteria should be considered when I should go for MIC?
I was thinking about growth curve. If we can decide about growth profiles of that organism, individual organism.
Yeah, it was done. It was done earlier.
Clear the picture about
exactly. It was done. Growth curve was performed. That was the first work which I did in the case of.
So is it needed growth curve as well as MIC? We can use both techniques for deciding the isolate.
Technically, it gives you better information when you perform growth curve also, because the organism should be able to survive. See, when the degradation of pH occurs mainly during the log phase, and when it reaches stationary phase, it starts to produce more metabolites. Okay, so it should be sustaining that log phase when it is degrading pH, because it is using it as carbon source. So it should have a good growth rate when it's degrading pH. Otherwise, there is no point. If the organisms die quickly, so the pH which is remaining in the media will be higher. So it should have a good growth rate when you are deciding MIC, at least for when you are doing biodegradation studies. In case of
Yeah,
other studies, it's
definitely.
different. In other places, it's different.
Because MIC will show the concentration they are degrading. But
Yeah,
what
yeah.
about their speed of growth, or we can say about their
Yeah, I didn't add the growth rate, but then yeah, the growth rate also was determined in this much amount of cells per ml in this much amount of this. So yeah, growth curve is also essential when you're degrading pH or any biodegradation, etc. When you're doing MIC in case of any other antibiotics or something, it's completely different at that time.
Okay, thank you, sir.
Yeah.
There are
So,
some questions in the
yeah,
chat.
yes, sir. Yes, sir. Yes. I can see.
Sir, Vivek, sir.
Yeah.
Sir. Sir, I want to ask you one thing.
Okay.
In these ring compounds, 4 ring compounds like a pyrene or trypsin, did you try with those substrates?
No, I have
Yeah.
not moved towards higher molecular like pyrene and all. I stick to low phenanthrene. Yes, I can move towards that, but I didn't focus because I wanted to focus how much
But
the
why I'm
2 ring or
asking,
3 ring compound was.
yes, why
Yeah,
I'm
yeah.
asking these
Yes.
ring cleavage mechanism is similar, means it will your isolates has potential to cleave
Yes.
that.
Exactly. Ring cleavage it can do. Yes,
And
yes.
if ring addition is going on, means that is more recalcitrant capacity in the environment.
Sorry, sir. I can't hear because there is some other interference.
Excuse me, everybody. Kindly mute yourself unless you are asking a question.
Rings are increasing more, means that recalcitrant to the environment.
Exactly. More rings, more recalcitrant. Exactly. That will be carried out further in further studies because that's essential.
Yeah. And one more thing, sir. In your consortium, all are whatever organisms are like any classify, like these are supporting bacteria. Yeah, these are the um, degrading molecules producing bacteria. Like that, you took any classified basis, like, all randomly you choose that.
Randomly, as in when I developed the consortia, I first checked with the individual bacteria whether they are able to degrade higher rate or not. Then only I developed them. So single organism should be able to degrade both the phenanthrene and naphthalene, and every single organism should be able to produce catechol, which was responsible. And then only these organisms can be used for the consortium development, not randomly. All the organisms should have technically same criteria, they should follow the same cleavage pathway. Otherwise, there will be some you can say conflict between the organisms. They might not give better efficiency of degradation. That's the reason the organisms which I chose for formation of consortia should be following the same degradation pathway and similar, not same, but in similar pathway. As you can see, there are 3 organisms which are chosen consortia. The genes which I found out, they were having 3 different genes for degradation, but the main mechanism of degradation was almost same. The naphthalene to catechol, phenanthrene to catechol, and then to TCR. So all should have the same pathway. Then it could give much better results as compared to the organisms which are randomly selected, which might have some subtractive, some negative effect also. So that's the reason. That's how I chose organisms.
Good, excellent, nice work.
Thank you.
Thank you.
Vivek,
Yes.
are you preserving the consortium?
Yes, actually I have preserved it, but actually during lockdown certain things changed. So I hope the organisms are still better. So individually they are there, but then I have to check with consortia once more.
Can I give you a suggestion?
Yes, sir. Yes, sir.
All of you who are working with microbes must preserve the cultures properly.
Yeah.
Because you will need them lifelong, those cultures.
Yes, sir.
So if possible,
important thing.
You submit the culture to a culture collection.
Right.
If deposit. Okay.
Yes, sir. Yes.
Because if you are having your culture in the fridge, they'll be lost in no time. Right?
Right.
So you have very precious cultures. You have done such a good work with phenanthrene and naphthalene.
Yes, sir.
So you cannot, isolate the cultures again.
Exactly.
So please, all of you, all the students, you preserve your cultures in a culture collection that is free of charge. If you give a culture to a culture collection, they'll not charge you any money.
Right.
They'll preserve the culture, provided it is right, free of charge for you.
Exactly.
Okay.
Exactly. Thank you, sir.
Very
Thank you.
important for all the students.
Yes, sir.
Okay. All right.
Yes, sir. So there are some questions.
Yeah, in the chat box. You want me to read them?
Yes, sir. Yes, sir. I can answer them if we have time.
Sure, sure. So the first one asks, what did you use in prototype model? Which contaminated water you used?
So I didn't get the wastewater from any sewage or something. The water which I used was general tap, which it was not also distilled water. It was tap water. So I checked for any other heavy metals or anything. So my main focus was in the presence of any other water source like wastewater. The pH which I add should be degraded. Later on, the studies can be carried out for other contaminants which are affecting the growth. See, degradation rate was very low. I would have checked for different components. So I definitely checked for pH and pH and other things like any other organic sources are there, but there was minimum source from wastewater which I chose. Only the organic compounds were pH, that is phenanthrene and naphthalene. I didn't check for heavy metals, and I also didn't choose any other wastewater like sewage water, which might contain more heavy amount of impurities. So my study was basic only till known and confined environment, and the water which I know, which may contain these compounds and these minerals. Apart from that, I didn't go for any further thing because this might have taken long time and more studies to be done. So I focused on just wastewater from the tap water, which might not contain all so much amount of impurities. So this was the water which I used along with organic carbon source, that is only phenanthrene actually.
Thank you very much.
This was used in my, yeah.
And now I would request you, sir, that kindly type your mail in the chat box so everybody can address their questions to you.
Sure.
We can have one more question, one last question, I guess.
Sure, sir.
Anybody wants to ask?
Here.
Yes, sir. Yes.
Please.
Very nice presentation.
Thank you, sir.
You were starting with the development, microbiology, and also.
Yeah.
Somehow I was unable to much biochemical and molecular evidences associated with degradation. The future perspective or
cracking.
Conclusion, if you can talk.
So sorry, actually voice is cracking, so I couldn't hear the words.
I was actually wanting to know any biochemical or molecular evidences.
Okay, evidences. Okay, so about this, I didn't do any. So I wanted to actually do some MS studies also, GCMS studies, where I could find out what are the metabolites or the molecules formed during the degradation. So unfortunately, whenever I used to do GC studies and the only two components which were there in my degradation was either catechol, salicylic acid, or the phenanthrene and naphthalene. Apart from that, everything was getting completely degraded. So main two components which I found out were and catechol, major was catechol. Hence, I could say that it was metacleavage pathway. Otherwise, it would have been very difficult to find out what are the metabolites. So mostly, the pH was getting degraded clean, and salicylic acid and the catechol were only the major components which was found out during metabolic studies.
Just to conclude, how was what is the novelty of study with reference to available literature? Is it a congestion,
novelty,
faster degradation,
Is it the metabolites which you get at the end?
So novelty of, if I can say novelty of my study, then first novelty will be the use of agromyces in my study. No, agromyces was never studied even individually or as a consortia. Second will be use of more organisms than 2 to 3 organisms in a consortia, which were giving the good results. Then the amount of the concentration of pH which I used was extreme. That is, I would say extreme because it is around 2000 ppm. Then the bioreactor which I used was quite a simple and lab scale bioreactor, where I could get degradation at good rate, at limited amount of not only just cost but also in all the sources which can be easily available in lab. So any student who is wanted to work on degradation studies can use these sources, these chemicals, and this kind of bioreactor for their own studies. So this I can say.
Just to get a reply from you once again. With
Yeah.
reference to bioreactor, what was the means? How unique it was? How you came to the conclusion that this is what the bioreactor is required for your studies?
The thing is, I actually wanted to, for bioreactor studies, I wanted to have a bigger bioreactor, better bioreactor with all the ports and all the pH ports. You have temperature probes, all these things which I wanted, but the cost was going very high. Plus, also I developed a bioreactor which was having all the biofilm beds. Then I wanted to add more things, but then some way the bioreactor was not getting constructed properly. So I thought of developing a simple bioreactor which will have necessary components for introducing the media, for introducing the pH, for introducing the NaOH or HCl if I wanted to change the pH, and also the filters which I could add for stopping the contaminants for entering the bioreactor. And also I wanted to add those glass slides where biofilm can be found. So it and also introduction should not be very hassled. The air also which I am added inside for mixing of the mixing of the media should be done easily, because the time required should be less for all these things than the actual degradation process. And hence I chose this bioreactor which I designed. Should be quite laboratory friendly, should be budget friendly, and should be easy to operate, and won't take much amount of time, but it would also have a contained environment inside which I can perform my studies, and all the necessary formation, biosurfactant formation, biofilm formation can be done by the bacteria, which you cannot do inside a mask or any other container which you use. That's how I chose the bioreactor for my study.
Wonderful, wonderful. Very good work. Congratulations.
Thank you. Thank you, sir.
Thank you, Vivek, sir. Thank you.
Thank you, sir.
Thank you, Alami, sir, for asking a question. Everybody, thank you very much. And if you have any more questions, kindly direct it to Dr. Vivek's mailbox mentioned in the chat box. And in the interest of time, I think we will close now. Thank you, Vivek, sir, once again. It was
Thank you very much.
Presentation.
Thank you, everyone. Thank you very much.
Thank you, respected Deshmukh, sir, for providing us this platform to connect. And it was very nice brainstorming. Have a nice weekend, everybody. See you next Sunday.
Congratulations and thank you.
Thank you very much.